Okay, good day everyone. So my name is LEE ZHI XUAN. Okay, this video I will explain about the organelles, the last organelle cytoskeleton. Okay, so before we go to the further of the detail of cytoskeleton, now we go to the structure of the cytoskeleton. So cytoskeleton is, in the short terms, it is like a bone to the cells. To the cells, meaning that our organelles that you learned before, like mitochondria, ribosome, nucleus, all that, why they can stay on the cells? Because they are supported by the cytoskeleton. If there are no cytoskeleton, meaning that that organelles is easily to move to anywhere. Maybe they can, they will akan they will bertumbung kepada antara satu sama lain, which will damage the organelles lah. So if we want to fix the organelles place, or we need to make a movement of the organelle, like the budding off of the endoplasmic reticulum to the Golgi apparatus, and also the budding off of the Golgi apparatus by using the secretory vesicle, like that. So if you want to do this, cytoskeleton play the major role to it. So what is the cytoskeleton? So normally cytoskeleton is the fiber network to extend your throughout the cytoplasm. So as I mentioned before, it is the, you can say that it is like act as a bone in our body. So in cells, the bone is we call as cytoskeleton. So if you see this diagram, so this diagram is under the one of the light microscope, fluorescence microscope. So you see this diagram. So all of these, you can see that all of these is we call as cytoskeleton. So all of these is support our organelles one. So you can see this blue color, this one is nucleus lah. So you can see all of them, any green color, orange color, all of these is cytoskeleton. So normally is to provide, the main function is to provide mechanical support and maintain the cell shape. And also, cytoskeleton also provide encourage for many organelles and also enzymes. So meaning that they support the organelles, so that the organelles will not move easily. They just retain the organelles at the one place only. Or maybe the organelles want to move, they need to with the help of the cytoskeleton. And cytoskeleton normally is more dynamic than the animal skeleton lah. Why? Because it can dismantle, meaning that it can disseminate or hilang in one part of the cell and be assembled in new location to change the cell shape. Meaning that cytoskeleton, although it acts as the bone in the cell, but more dynamic, meaning that it can easily to be disappear and then form again. That's why that we, especially the animal cell shape is different to each other. Although the cell type is same, but the cell shape may be different because of this cytoskeleton. Compared to animal skeleton, that bone you cannot even change the shape easily. When your bone is like this shape, then it's the shape. It's the 10 cm, then 10 cm. You cannot simply want to change to 12 cm, 13 cm, or you want to shorten it. It's not easily. Very not easily, because bone is very strong, right? It is very hard and strong. So compared to cytoskeleton, so it is more dynamic lah, compared to the animal skeleton. And also when you see cytoskeleton, so normally cytoskeleton, if you want to move the organelles, any organelle or any substance, especially vesicle that I mentioned before, like the secretory vesicle, transport vesicle, or any enzyme, amino acid that be produced within the cell, and this cell, this enzyme, these organelles need to transport, move around surrounding the cell. They need the help of the cytoskeleton with the motor protein. So you can see this diagram. So this red color is the motor protein. So motor protein, what is motor protein? Motor protein is the very powerful protein molecule that can bind to the any organelles or vesicle within the cell, so that it can provide the movement of the vesicle from one part to another part, or even though they want to do the exocytosis lah. So normally cytoskeleton also involve the cell motility with the help of the motor protein. So these work together so that whole cell move along fiber outside the cell. Allow the whole cell move along fiber outside the cell. So the vesicle and organelles will use this motor protein to fit to walk in the cell. There's dissentation along a thread provided by cytoskeleton. So you can see this motor protein is like a walking. Like it is like O1. You can imagine it is the human that he bring the things like this motor protein is one human lah. They brings the things at their shoulder, and then they walk along the microtubule. So microtubule, you can imagine is like a pathway lah, or the jalan. It is a jalan that can make the vesicle, angkat the vesicle, bring the vesicle to transport the vesicle from one part to another part. So that is the motor skeleton, one of the function of the cytoskeleton that you can write lah. Okay, so this is just a basic only. So in cytoskeleton, there are three major types of the cytoskeleton that can be formed, which is the three major fiber lah. As I mentioned before, cytoskeleton is made up of composed of the fiber, so protein fiber. So there are three types of fiber: microtubule, microfilament, and also intermediate filament. So we see it one by one lah. Okay, so about the microtubule, so you can see first one we see about microtubule. So microtubule is the major fiber. Major protein fiber that compose to make up the cytoskeleton in the cell. So you can see that microtubule is quite thick. It is a thick. You can say that it is a hollow tube to make from the globular protein called tubulin. So tubulin is the protein molecule lah. So when there are many tubulin protein combined together, joined together, they will form the microtubule fiber. So normally this one microtubule will go in length by adding many tubulin lah. So also can be assembled and used to use tubulin to build microtubule. And then the two ends of the microtubule are different to each other due to tubulin acts as diamond. One end can accumulate or release at much higher rate than other. It is important in growing and shrinking during cellular activity. So this is just one of the characteristics of the microtubule. So you can see this. Normally, tubulin is function as the dimer one, meaning that there are different type of the protein lah, different type of tubulin. This one is, you see this yellow color is alpha tubulin. This one is beta tubulin. So they need to arrange in the dimer. So normally if you see this at one end can accumulate or release at much higher rate than other one. So this is very important to growing and shrinking during cellular activities. So when the microtubule want to grow, when your cells need to grow, need to increase its size, microtubule is this tubulin structure is very important. If you imagine all the cells, all the microtubule, the tubulin structure is arranged badly, there are no space. If there are additional tubulin want to add, if there are no space, so no addition process occur, and the microtubule will retain their shape and forever lah. They cannot change the shape anymore. So this is very important structure characteristic even between in the microtubule for the growing and shrinking activities in the cells. Okay, and this one is the general function of the microtubule. So the main function is to form cytoskeleton. This is the major component of the cytoskeleton. To form the whole cytoskeleton. And then of course, if therefore for cytoskeleton, as I mentioned, it provides the mechanical support and also give the shape to the cell. And also divide cytoplasm into compartments to isolate specialized enzyme. So this microtubule able to isolate the enzyme one. And then another one is concentrate for the movement of cilia and flagella. So this one, cilia and flagella, is normally special for animal cell. If you still remember that in animal cell, some of the animal cell containing cilia and flagella, but plant cell will not have lah. Normally, plant cell will not have. Only the animal cell, cilia and flagella. So normally cilia is like normally you can see in our nose. Our nose got the cilia, right? Our nose, our digestive system, all this containing cilia. And flagella normally in animal, the most important is sperm cell. Later we will see it more detail about the cilia and flagella. And then also number E is to pull chromosome or chromatic during mitosis and meiosis. Yeah, centriole is also one type of the microtubule. Microtubule also will form the centriole, so that it can play the role in the mitosis and meiosis process. And final one is serve as thread for intracellular movement of organelle or vesicle. So of course because it containing motor protein, with the help of motor protein, so that it can move the organelles and vesicle easily. Okay, this one as I mentioned centriole. So this is centrosome is one type of the microtubule formed in the animal cell. So microtubule, when they grow, can combine together to form centrosome that is located near nucleus. So microtubule is the one long hollow root structure. So when there are many microtubule combined together like this, so you can see this diagram. So this one is centriole, right? And we know that in animal cell normally we containing a pair of centriole. So when they combine together, they call as centrosome. A pair of centriole is equal to centrosome, the scientific terms. So when there are many microtubules combined together, joined together, they will form the centriole structure. So normally within the centrosome, it's a pair of centriole. Two centrioles are arranged perpendicular to each other. They arrange perpendicular to each other, like this. One is horizontal and one is vertical. One is vertical, one is horizontal. They will not do, both of them are neither horizontal or neither either vertical one. Both of them need to different direction. And so normally centriole is very small and hollows in either about the 0.5 to 0.5 micrometer long and 0.2 micrometer diameter. And number 4, this one is very important that I need to mention, is the 9 plus 0. So the arrangement of the microtubule in the centriole is 9 plus 0. So you can see this diagram more diagram. You can see that each side containing 3 microtubule. You can see 1, 2, 3. 1, 2, 3. So 9 plus 0 is there are 9 triplet microtubule arranged together to form one microtubule, one centriole. You can see there are 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplet microtubule joined together to form one centriole. What about 0? 0 is the center. You can see there are no microtubule in the center one. So that is why they call as 9 plus 0, which is the 9 set of the triplet microtubule arranged in a ring form to form the one central structure. So normally it's found in the animal cell, and the major function, as I mentioned, is play the major role in cell division process, mitosis process and meiosis process. And another one is about the cilia and also flagella. So cilia and flagella is also made up from the microtubule. So you can see this is the structure of the flagellum and also cilium. So normally this one is more focused on the flagellum. Okay, so what I need to tell you is that if you see this now, so this one is the cilium. When you enlarge the cilium structure under the electron microscope, you can see it is like that one. All of these, the initial part, all of these is microtubule. And the other part, you see this part is the cell. The cell structure, or you can see this all of these is the cytoplasm one. This part is the cytoplasm, and this one, the longer one, is the cilium or flagella. So this one is the case is cilium. So what what I need to focus is that when you cut down the cilium or flagella, when you cut down it, you see the cross section of the cilium is different from the microtubule inside the cell one. Microtubule inside the cell will have the similar structure in the central one, which is 9 plus 0, meaning that the 9 outlet doublet of a cilia or extend into the body. Each triplet is connected to the next by non-tubulin protein. Yeah, so normally I need to focus is that it is triplet, 9 plus 0 triplet, similar to the central one. Inside the body, within the body, the microtubule structure is 9 plus 0. But when you go to the cilium or flagellum, the structure, the cross-section structure is different. It will become 9 plus 2. And this 9 is double. The microtubule only double only. Each set only containing 2 microtubule. Compared to this one containing 3 microtubule. Or in this one, for the flagellum or cilia, they are containing only 2 microtubule. And 9 plus 2, this 2 is at the center. In the center, there are 2 microtubule at there. That is why that 9 plus 2. So this is very important that I need to mention. The doublet also attach motor protein. So normally you can see this structure, there are many protein at there. There are many motor protein at there. You compare to the triplet one, so normally when they extend into the cell, they will become the triplet, they will become the hollow zone. This one is not hollow zone. You can see there are 3 lines, right? 3 lines. This line and this line is the triplet like this, and then you can see at the middle line is these 2. We call of these 2. And once you go to here, you can see that at the middle line is missing. Because the middle already does not containing the microtubule. There is no microtubule at all. Okay. So this is the different structure of the cilia and flagellum. So when you see this one is more detailed. When you see this is cilia or flagellum, you can see that only 9 plus 2 like that. Okay, so this one is just show you how the cilia and flagella move. The motion of the cilia and flagellum. So normally flagella is the structure, is a tail-like structure that make the cell can be easily moved from one place to another place. So normally one of the main example in animal is sperm cell. Sperm cell they're containing the tail, which is called as flagellum. So this flagellum is the place to make the cell can swim from one part to another part. And another one is the cilia. So normally cilia they cannot move. The main function is not a movement of the cell. It is just the cilia just that it can provide help the cell to discover to beating the something lah, either something lah. Okay, so that's all about the cilia and flagella. So that's all about the microtubule lah. Now we go to microfilament. So microfilament is the cytoskeleton, another type of cytoskeleton which is more thinner to microtubule, and it is not a major component to form the cytoskeleton. Only the certain part of the organ, certain part of the body containing microfilament, not all part of the body containing one. So one of the example is actin filament. So microfilament are thin solid tube, also called actin filament. This one is made from actin protein. Compared to microtubule is tubulin protein, tubulin globular protein. This one is formed from the globular actin protein. Okay, and normally the arrangement of the microfilament is they are intertwined strand of the actin, meaning that they are twisted together. Compared to microtubule is like a tube. You can imagine it is like a pipe, one pipe, and it is hollow. And for this one, Microfilament, they are not arranged in the hollow row, one tube only. They arranged in the twisted mode. This one is the twisted mode, and the arrangement is like that lah. You can see. So normally, can form structural network when certain protein bind along the side of the microfilament to allow next filament extend advance. So normally, these microfilament are found in the muscle tissue, especially skeletal muscle. Skeletal muscle tissue containing many is one of the major component in skeletal muscle is microfilament, actin filament. Actin filament. Okay. So normally the function of the microfilament is play the major role to bear the tension or pulling force. And then another one is to support shape of the cell. And another one is to nutrient absorbing intestinal cell made up of core of microvillus of the finger-like projection to increase the surface area projection. So this one is the one of the characteristic. Finger-like projection in the small intestine is due to this microfilament. So I give you the you will see here this one. You will see this is the small intestine microscope structure. So you can see that if you still remember that in your SPM you learn about the small intestine absorption, right? You know that small intestine containing many finger-like projection like this one. Many finger-like projection like this to increase the absorption rate. So all these finger-like projection actually is caused by this microfilament, actin filament. Know that this actin filament can form many tube or many line, can form many microvillus lah. So they can form many microvillus to make the small intestine have the finger-like projection to increase the surface area of adhesion lah, so that more nutrient able to be absorbed in the small intestine. So this is one of the location that our small intestine containing microfilament. So not only small intestine, the major one is also in muscle, especially skeletal muscle. It plays the role in skeletal muscle contraction. So this topic later you will learn back in semester 2. Yeah, you will learn back again. But for now you don't need to know lah. You just want to know that when this microfilament, actin filament, will cooperate with the thick filament, which is another one protein we call as myosin, where they interact to each other, they will form make our muscle contract. The mechanism of the muscle contraction lah. And another one in plant cell, normally the actin protein will interact to contribute cytoplasmic swimming process, which can make the cell can be circular flow in the cell lah. So this one is the one of the transport in the cell that you also will learn in semester 2. Yeah. So this donkey and E is the further topic that you will learn in semester 2 lah. And I just want to tell you that the major component why they can carry out this function due to this structure. If there are no this structure, then this two function cannot carry out. Yeah. And this one is I already mentioned. This one is the structure in the muscle. What is we call as sarcoma. But this one you don't need to know now. I just show you only. If you see this yellow color is the actin filament, right? And this one, blue color is the what we call as myosin filament or thick filament. So this process, these two structure is the major component of the muscle cell, major protein component of the muscle cell that play the role in muscle contraction. So they will play the role in muscle contraction and using this kind of structure. So this one you will learn back in semester 2. I just show you only that one of the function and this one is the cytoplasmic swimming in plant cell where the actin protein interaction. So I mentioned before, this one you also will learn in semester 2. So you don't need to know more detail now. You just want to know that in plant cell, the actin protein interaction will cause the circular flow inside the cell. So that the water, the mineral, not water, the organic solution, sucrose can be moved within the cell easily, and contribute the transportation of the organic nutrient, especially sucrose, from one cell to another cell. Okay. Okay, the final one is the intermediate filament. Okay, intermediate filament is the thinnest filament. Sorry. Okay. It is the thinnest filament cytoskeleton in the cytoskeleton lah. So among the three structure, interfilament is the very thin. You can see the length or the diameter of the filament is only 8 to 12 nanometer. It is nanometer. Compared to the microtubule and also microfilament, they are micrometer. They still micro, but this one already up smaller to nanometer. This one is already, even using the electron microscope, you only can see very good. Even very thin. So this one is the intermediate filament. So normally it is made up the fibrous protein. The different. Compared to microtubule and microfilament, both of them are made from globular protein. You still remember these two, the classification of the protein, globular protein and also fibrous protein. Both of them are different. This one, intermediate filament, are fibrous protein. So normally, many fibrous protein, one of the fibrous protein is keratin. So this one fibrous protein, you will still remember that you learned in chapter 1. Fibrous protein normally is the secondary structure. Not secondary structure. They're containing many secondary structure. Normally it's a helical, coil, or beta sheet structure. So normally it's coiled together. That is why that you can see, although it looks like a tube, but you will see in detail, actually they are coiled together to form the tube structure like that, filament structure like that. Normally the diameter is longer than microfilament, but shorter than microtubule. And it only found in some of the animal cells, especially in vertebrata. So not all the part of the cell or the body containing this kind of filament. And normally it is more permanent feature than microfilament and microtubule, meaning that this filament they can provide very long support towards something. Compared to microfilament and microtubule, they not provide the long support one. They only provide the support when necessary only. But for this one, they normally can provide very long support lah. And they can be deassembled and reassembled in various parts of the cell. And even the cell die, it often persists. So this is one of the characteristic. So that is why that when the people die, keratin, one of the keratin in our body is hair, our bamboo hair. So even though you are died, but your hair normally is still maintained at that. Normally you see the movie, even though you are already died many days, or even though it's several months, but your hair still remains at that. Your body is already becoming already decay, but your hair normally still maintained at that. No decaying at all. The decaying is very slow. That is why one of the characteristics within the intermediate filament. The function of the intermediate filament, so normally, the major role is to provide the shape of the cell and fixing the organelles in a cell. They provide the fixing position of the organelles and synthesis the nuclear lamina. Still remember what is nuclear lamina? If you already forget, please refer to the nucleus video. Another final one is supporting the cell shape to carry out specific functions. So supporting the intestinal microvilus. So that's why that when you see this diagram, you can see that this is actin filament, and below the actin filament there are many intermediate filaments. So they provide the support to the microfilament like that. Okay. Okay. So that's all for this video. So hopefully you guys can learn something in this video about the structure and also function of the skeleton, cytoskeleton. So thank you for your watching and bye.